Was yours.
All right, thank you very much. All right, thanks everyone for coming. I'm going to be telling you a little bit about a simple abstract model that some students have been working on for the last couple of years, and can explain what our approach is here to trying to take a really simplistic abstract approach to modeling development, and then trying to explore what some biological phenomena such as exaptations could be explored within that. So one question you may want to ask is, why should we try to study development with abstract models? Well, I think a lot of us are very interested in development as a very adaptive and complex process. And so to be able to study it beyond just models of very specific organisms, it can be helpful to try to make it more abstract in general. And there's also an opportunity here where we can try to look at large complex processes such as learning, development, and evolution, and try to see interactions between those whenever we can build these. So that gives you a little motivation for that work. Specifically talking about exaptations. So exaptations are examples where we have evolution repurposing things. There's some documented cases. One really cool one is this concept of an antifreeze protein that's produced in an Antarctic fish that originally came from genes that produced a pancreatic compound that ended up being repurposed. So this is where evolution was tinkering and found something that it can do. Now, in general, these are very hard to study. We do have a few examples, but they're exceptionally hard to predict. And they might be much more common than we realize, but we don't have great ways to approach it. Perhaps even more challenging would be a concept of a developmental exaptation. And we do know that from a lot of work in Evo-Devo, that there are reuse of components, such as oxygenes. And so we suspect that there's probably lots of cases where developmental processes are being repurposed and reused. But again, this becomes even more difficult because of the time scale and trying to observe something actively through development to understand the degree of those processes. So our goal then is to use simulation models to be able to try to simplify and clarify conceptually what this could look like, and then try to do some investigations to understand things that could be very difficult to otherwise access. So trying to build off of a specific definition here and trying to map the model to it. I'll just read a section of this. So Chipman, 2021, define this as a process through which a pre-existing characteristic of a developmental program can be used to facilitate evolutionary changes in development. And so we'll take a look at how we can take this simple model and apply that concept to it. And so we're going to build a model, look to evolve populations, try to see, can we detect this type of thing occurring, and then study the mutations that could make that be possible, look at trying to replay the evolutionary tape and see if we can get some insight into the evolutionary dynamics. So the model that we're using has to do with an organism moving through a landscape. And so what we are trying to model here is that over a lifetime, as an organism is developing, there can be changes in the phenotype, which those could be physical characteristics, but those could also be behavior. So perhaps traits that you've learned during your lifetime. And so what we're trying to do is model that state, or that collection of behaviors, that landscape, as states where an agent can move from one state to another, and we can represent that concept of being in one position versus another as having different learned traits up to that point, which would then influence how fit it is. So this is a little just kind of depiction here. We can have something kind of in a discrete space like a graph. These have been done with NK fitness landscapes and some older models, but we're going to talk about a very specific toy landscape today. But the general idea is that as we're moving through this landscape, there's going to be a fitness associated with each location, and we might be going up or down, or in our case, we're also going to have the potential that as our lifetime, as we're exploring, we might also have a action that says, let's do the last thing that we just did, to kind of represent this idea of a developmental process where kind of one thing cascades into the next, which will be very key to how this whole process is going to work together. And so the genome of our agent, they're going to start at one particular location consistently. The genome is going to encode what type of learning action we're taking here. So what kind of moving up or moving down or repeating a pre-existing step. And so this is kind of taking a explore, exploit, continue approach to these actions. So in terms of the model, what we're going to use, we're going to have an indirect encoding scheme. So starting at the top, the overall organism's genome here is going to be these three blocks, B1, B2, and B1. You'll notice that B1 there is used more than once, so it's potential for that one gene to encode multiple parts of how the learning is happening over the lifetime. There is a block bank, so this is kind of the genes that maybe become active if they're in that developmental program at the top, and so that can potentially have mutations in it as well. Each of those blocks in the block bank has an encoding. So you can see B1, it has that starting with a climbing step and then a series of repeating steps, whereas B2 has a falling step followed by repeating, which would make it continue to fall. And so we would go and take that indirect encoding. The top would then get translated into a climbing step followed by a sequence of repeat. If we look at B2, that would then get translated likewise into its block and then to the third one. And so that sequence at the bottom would be all the steps that the agent would take during its lifetime. And then we'll evaluate how fit it is at the very end. And we can run evolution to try to evolve effective programs. So as a preliminary investigation, we wanted to try to set up a situation that was ideal to observe exaptations of this developmental process taking place. And we wanted to make a landscape that would be relatively difficult to solve without having kind of some special conditions occur, and then wanted to evolve it and see if we could assess how that whole process was working. So this is the landscape. Each node is just connected to kind of one or two, three other nodes, depending on the junction where it's at. And the fitness here is relatively proportional to height. So all agents are going to start at node A. And if you look at the graph here, it's clear that the only way you're going to be able to get to node D in the upper right there is by basically going upward, downward, and then upward again. So in order to make this challenging, we're going to start off by initializing the population. In a lot of ways, we're kind of putting a presumption here that something has already evolved, and we want to see if it can be repurposed in another way. So we are going to initialize the population to only have a single block. It can potentially have more than one block, but for the start, it's going to have one block. It's going to have a climbing step followed by a number of repeating steps. And so any agent in this initial population is just going to climb all the way up to this node C. And as you can see, in order to get to node D, there needs to be some changes. We're going to have this potential to actually go down before we go back up. And hopefully we can do that before our lifetime runs out, which there's enough space for us to be able to do that. So in order for that to occur, what we're trying to look at here is, again, mapping this definition. What are these pre-existing characteristics of a developmental program that can be used to allow change? And in this case, what we're really looking at is that long sequence of repeated steps that we have in block one. So having a whole number of repeats in a row makes it possible for us to have a radical change in behavior with a relatively small mutation. And that's possible because we have this indirect encoding scheme and this kind of cascading process. And so on the left here, you can see that there's three different types of mutations that we really see as necessary then in order to be able to reach that global optima. And in our definition and how we're mapping this, we see this as an exaptation. So the first thing is looking back to our goal here, right? We want to try to get up to node D, which means there has to be some kind of mix of climbing and falling in our genome. And so we really need to have some kind of block copying. So we need to be able to have kind of two different genes, one that can be a copy of the other and then needs to be changed in some way so that it's not strictly climbing. So we can have some falling and some climbing. And then we also need to put that into the developmental program as one of the blocks that is executed during the lifetime. So then a hypothetical way that this could happen is you could imagine having that first step in block one be changed to a falling step. And then we could have this sequence of climbing up, falling down, and then climbing up again. So this was just an idea of what could happen. We wanted to see what evolution would do. So We ran this and consistently were able to get agents that would hit the global max. And then that's where the fun began of trying to investigate, okay, what's going on? What kind of insights, what kind of analysis can we do? So one thing that we did was we took agents. So these are just mutating agents. These aren't performing crossover. So it's very easy to follow the lineage back to see where they came from. And we could also set things up to easily detect those three types of mutations that we mentioned being necessary, those block copying mutations, block function mutations, and the block introduction mutations. And we can see those are being spotted here. Don't worry about the height of the dots. Those are just in a line in the same color to make it easy. Those aren't going to be related to the fitness. And we can see that those conditions are happening. We're having those mutations occur before there is that spike in fitness where we reach the global optima. So can we get some insight further kind of beyond that? This is just kind of seeing that those mutations are happening. Some of them are happening a couple times before we get success. We can also go and look at the population statistics. So again, looking over evolutionary time, are we seeing these different conditions being present, right? Has this mutation that we expect to be necessary, does that exist in the population? What percentage? And so you can see that the lines are gradually climbing there. You can also see that while not everyone has, achieved each of those, once we have that population spike where we're hitting the global max, there's a quick increase of most of the population to have those necessary mutations to allow them to reach the global optima. Now, something else that we can do is we can use this effect or this approach called potentiation, something that was published last year at A-Life 2023, where we can try to replay the evolutionary tape and say, all right, let's take a generation at a particular point, or a population at a particular generation. Let's simulate evolution forward 20 generations. Let's do it 1,000 times for high fidelity. And then let's see how many times did that population reach the global fitness. So was there at least one agent that reached the global fitness? And what we can see here is that even though the best fitness kind of through that first 60 generations or so is pretty fixed, we can see that potentiation going up and down significantly, which is kind of interesting. Maybe there's a chance of it being higher. Once we have that one individual that hits a higher point, and in this case, this is a specific one we picked. We wanted to take a look at a run where we didn't immediately jump to the global fitness, but a big jump above. You can see the potentiation spiked as well in the pink. Okay? They just happen to line up on a similar scale, which means that even though we haven't hit the global fitness yet, there's a very good chance that we will be able to in the next 20 generations, and then it continues to progress upward from there. Now, to get a little more insight into that, we wanted to also look at, okay, there's a lot of noise here. There's some percentage of the potentiation going up and down. What if we were to take one individual on a successful lineage and actually just make a whole population of that individual and do that same effect of potentiation? But now we've just kind of cloned that one individual to say, how likely is this individual to be able to actually get that global maximum, treating it as a population? And what we saw here was that when we're looking at those kind of crucial mutations, almost each case where we had a crucial mutation show up, we saw an increase in potentiation. Now, not 100% of the time, but in a lot of these times, we did see that it goes up, which is kind of a cool effect of being able to have a sense of forecasting that, hey, we are actually increasing the odds by having some of these required conditions. Now, this was all made easier by setting up a model where we saw that we really needed to have these and being able to do this in a more tractable manner than to generalize, which is really where we want to go next with future work. But just to take a little bit deeper dive, if you want to know kind of what's going on with that specific agent, it ends up introducing a falling step at a particular place. It also kind of replaced an initial climbing step with a repeating step. Again, kind of the details of that you can look at more in the paper, but it kind of had this chance to get an exaptation, even though it didn't get to the global fitness because it did repurpose it, but then later on was able to get up to that global one. So what we'd really like to do is move beyond this kind of very artificially constructed toy landscape that made it easy to study this problem to more complex landscapes like NK landscapes, and especially dynamic NK landscapes where you have things changing, and be able to work on really generalizing this to be able to detect these types of conditions, be able to try to tease apart some of the difference that some of the trends that may be happening, and then really look to incorporate other aspects of interaction between the agent and the environment. So we don't just have those really simple steps, but we also might have things like looking at maybe a subset of the local environment or something about the agent's lifetime history. So in summary, we have been able to do a preliminary simulation of developmental exaptations on this toy landscape. Being able to trace the lineage and identify crucial mutations gave us insight into what was happening. Potentiation was a nice way for us to kind of see what was going on behind the scenes when the fitness wasn't really changing. And we think this sets us up for some nice opportunities for future work to generalize the model, gain more insights more broadly. Thank you all.